Hello everyone, my name is Rauf. I am a student in Tishki International University, Biology Education Department 4A. In this video, I want to talk about the replication and life cycle of viruses. Before we start, we have to know that viruses are not like our cells, or they are not like our other living organisms. Most of the times, they are called non-living particles because they do not have their own replication mechanism. They cannot replicate by themselves. What they do is that they infect our body cells, and they use the mechanisms inside the cells to replicate and make more of themselves. Okay. First of all, before we start, let's talk about the structure of viruses. The structure of virus mostly contains these ones. The head part, okay, which contains the genetic material as well. The head and genetic material. Okay. Another part that we have after that is the collar. We have a structure, a circular structure beneath the head, which is called the collar. And after that, we have the tails of the virus. The tail actually contains two parts. First of all is this one, and the second one is the tail fibers. Okay? This is the tail, and these are the tail fibers. These tails and tail fibers help the tail fibers help the virus to attach to the cells, and then the passing of the genetic material into the host cell. Okay, now let's talk about the life cycles, or let's say the replication cycles of the viruses. The viruses use two types of replications. One is called lytic cycle, and the other one is called lysogenic cycle. Okay. We are going to talk about lytic cycle first, okay? We are going to talk about the lytic cycle first. Lytic cycle. In the lytic cycle, we have 5 steps. First of all, the virus has to attach to a host cell. Okay, this is called the attachment step. Okay. This is our host cell. Let's draw our virus as well. Let's say this is our virus. Okay, let's imagine that this is our virus. With its genetic material. This first step is called attachment. This step is very important for a virus, because if the virus cannot attach to the cell, it cannot inject its genetic material, and it cannot use the cell's mechanisms to replicate. Okay. The second step that we have is called entry, where or let's say injection, where the virus injects its genetic material inside the host. Okay. The virus injects its genetic material into the cell, and it's called entry. Okay. After that, we have another step where the virus uses the host cell to replicate inside and make more copies of itself. We are going to just draw one of the viruses, like an illustration, but actually the host cell will be filled with viruses. Okay. First of all, it will only make the parts separately. For example, the head and the tail separately, the tail fiber separately, and the genetic material separately. Okay. This step is called replication. they have to come together to make a fully grown virus, or let's say a fully developed virus. And this step is called assembly. Before that, assembly. Assembly, where the virus becomes complete with its all parts attached to each other. And the last part that we have is that this cell will be broken for the release of the virus, and the virus will come outside. Here the virus will meet the cell membrane to be broken and escape outside. The viruses that do this type of life cycle, they are virulent because they cause damage, they destroy our cells, and they cause diseases and sicknesses. The second replication cycle that we have is called lysogenic cycle. What makes this cycle different from the other one, from the lytic cycle, is that in the lytic cycle, our virus, or let's say the virus, destroyed our cells and made a lot of copies instantly. Okay. After the injection, it made the replication and used the mechanisms to make more copies, and by destroying the cell, it exists the cell. But this time, the lysogenic is different. How? After the injection, the virus DNA stays latent inside the cell. I will explain further. In the previous one, as we said, I destroyed the cell, but this time the other one was 5 steps, and this life cycle is only 4 steps. After the virus attached to the cell, this is the host cell again, and this is our virus. The first step, as we said, is called attachment as well. Attachment. After it is attached itself to the host cell, it will inject the genetic material or its genetic material to the cell, just like the previous cycle. And this time, let's use a different color. This time, with the genetic material of the virus, we have the genetic material of the host cell again, as well. What's different from the previous life cycle is that this genetic material of the virus will fuse with the genetic material of the DNA of the host cell, or let's say the DNA of the host cell, and it will stay latent. It will not replicate, it will not start replicating, it will not cause any damage or disease. Okay? And it will stay with the host cell for a long while. This step is called injection. Injection. And after that, the next step is the genetic material of the host cell, and this is the genetic material of the virus. It will stay like this until the cell decides to replicate and undergo mitosis. Okay. The cell will make more copies of itself, okay? And they are all containing the genetic material of the host cell and the genetic material of the virus together. of these viruses come out, they will start. Okay, let's just write. This is called latency. Okay, and this is the replication of the cells. What's different that after these viruses became active again, they are latent here. They do not produce or they do not replicate. But after they become active, they will activate inside all copies of this cell as in the same time. Okay, when the conditions are favorable for the virus, they will all become activated again, and they will start replicating, assembly, and destruction of the cells all at once. And that is what makes the two life cycles of this viral replication different. One was destroying the cell directly, and the other one is latency. And that's it for today. Thank you for watching.